Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Today, we're going to talk about the modern synthesis. Now, for whatever reason, this concept is often left out in most introductory textbooks. I don't understand why. It's incredibly important. We do learn about evolution by natural selection. We learn about Darwin's theory. We learn about Gregor Mendel and heredity. And we tell you that individuals can't evolve, but a population does. And it's really this idea of natural selection, population level thinking, and our knowledge of genetics and heredity that we have to wrap up all into one theory called the modern synthesis. And this is our current understanding of evolution, and it's very important. And so let's talk about this. Our learning objective today, it's going to be pretty straightforward, right? We're going to explain how the modern synthesis reconciles Darwin's theory of evolution by natural selection with our knowledge of heredity, starting with Gregor Mendel and population genetics. So we're going to come up with this synthesis. We're going to combine all of these different things like heredity, population level thinking, and evolution all into one cohesive theory. And let's not forget that there's this theme that's running through this course. Nobody works in isolation. Everybody is influenced by other people. Even Darwin was influenced by Lyell and Malthus. And here's Sir Isaac Newton. If I have seen further, it is by standing on the shoulders of giants. His work built on the knowledge of other people. And nowhere is this more true than the modern synthesis. It wasn't done by one person. It was done by a lot of people, each making significant contributions. So here we are, 1859. Darwin is basically starting to revolutionize science with his theory of evolution by natural selection, because that's when he published on the origin of species. And he had 500 pages. I mean, he had 20 years of data going back to 1831. I guess that's more than 20 years, isn't it? It's a lot of data, right? I mean, he had lots of information to support his theory. But there were gaps in our knowledge. We'll talk about those. So after the 1860s and into the 1890s, his theory began to wane because of gaps in our knowledge. However, by the early 1900s, evolution became once again popular. I should say evolution by natural selection. And like I said, it waned because we didn't know enough about how things worked. And theories receive valid scientific criticisms. I mean, I know that evolution is still criticized today, but for the wrong reasons. But when it was published, there were valid scientific criticisms. And there were gaps in our knowledge. We didn't know about genetics. We didn't know about heredity. We didn't know about the Earth's age. And we didn't have a good definition for a species. So one of the big ones was Darwin said, hey, the Earth is old. He believed Lyell and his principles of geology. And but not everybody believed the Earth was old. We didn't have the knowledge to do that. So, if the Earth isn't old enough, how are you going to have this slow change over millions of years? So that was a valid criticism. Another one was in genetics and heredity. Darwin proposed that there were variation in a population and that these variations were heritable. You could pass them on to the next generation. Well, if you don't know your source of variation in a population, if you run out of variation, evolution stops. And at the time, this idea of blending inheritance was popular. Yellow and blue make green. Well, if you have a little bit of blue and you keep adding green to it, it gets less and less blue until it's all swamped out and all you have is green. And it was thought that that's how traits were passed on. So, hey, where's your variation coming from? And any trait that did show up would quickly get swamped out. So these are the problems that Darwin faced in his theory of evolution by natural selection. But science to the rescue, right? Hey, promoting scientific theories and move science forward. This is an important point, right? Somebody puts out a theory like evolution by natural selection. Guess what? Somebody's going to go test it. Somebody's going to either push it forward, try to disprove it, fill the gaps in our knowledge. And that's exactly what happened. Let's take a look at this. And importantly, science is repeatable. We can repeat all of this stuff. It's fantastic. The age of the Earth. For a long time, the Earth was thought to be fairly young. We had no reason to believe it was as old as it is. For whatever reasons, a lot of people thought the Earth was mere thousands of years old. I would love to live 1,000 years. That's a very long time. But in the 1820s, Lyell published, his principles of geology with this concept of uniformitarianism. And he realized that the Earth is likely millions of years old. That was important. And then, in the 1860s and 1890s, Lord Kelvin, William Thomson, he's, famous for the Kelvin scale and studied thermodynamics. And he said, well, we know the size of the Earth. It's a body of rock that we can calculate how long it would take for it to cool off. It's still got a molten core. They can see lava coming out of volcanoes. So it's still hot in the middle. Based on the size of the Earth and how long it would take something this big to cool off, he predicted the Earth was 100 million years old. That's pretty old, considering we didn't know anything about radioactivity. Of course, in the early 1900s, the Curies discovered radioactivity. Now, it wasn't for another 50 years until 1956 when Claire Patterson, who had been spending a long time studying radioisotopes and was able to finally use various radioisotopes and this radiometric dating to determine the age of the Earth, and he figured out that it was 4.6 billion years old. That is far older than anybody ever thought it would be. It was ancient. So there we have plenty of time for evolutionary processes to occur. And that timescale is something I'll talk about in my next lecture on the evidence of evolution by natural selection. Here it is, the other one. Here are these Anolis cuvieri. These are crown anoles from Puerto Rico. Do you see the different colors? There's variation in a population, right? So nobody knew these sources of variation. They didn't know about mutations, something we'll come back to. And we didn't know how they were passed on. So if you didn't have new sources of variation, evolution stops. People believed in blending inheritance. Yellow and blue make green. We already talked about that. So these are questions that had to be solved. So we know that Claire Patterson figured out the age of the Earth. Now let's move on to the biology part and let's get into the modern synthesis. Remember, it takes a lot of people to get us to where we're at. A lot of people. Julian Huxley, Gregor Mendel, Thomas H. Morgan, Ronald Fisher, J.B.S. Haldane, Sewell Wright, Ernst Mayr, and of course Theo Dobzhansky. And it was about the 1940s that we were able to take all of their work, including Darwin's. I know I didn't put Darwin in there, but it took all of their work on genetics, heredity, and population genetics and a species and a definition of species to create the modern synthesis. So this is by the 1940s. This is 80 years after Darwin published it. So let's talk about these contributions. Gregor Mendel published his papers on heredity in the 1860s. They would have really helped Darwin, but he's Austrian, right? They were in German. He speaks English. They never just got together. Gregor Mendel, of course, is known as a father of modern genetics because he came up with the first rules of heredity. Now, they were a bit simple, and he got lucky. And as most of you know by now, Thomas Hunt Morgan is my favorite person as the founder of modern genetics, not Gregor Mendel. But Gregor Mendel's biggest contribution was he disproved blending inheritance. That whole yellow and blue make green. So the problem with blending inheritance is that if you get a little bit of a variation, it would quickly get swamped out through subsequent generations. Gregor Mendel showed particulate inheritance. A gene is actually a thing that can be passed on from one generation to the next. So that's particulate inheritance. That was enormously important because if a gene is an actual thing and it works, it can quickly get magnified in a population over time. It won't get blended out. It can actually grow with the population. So if you've got a gene that works, right, or a variation of a gene, then it can quickly spread through a population in several generations. So these traits are genes. This is a heritable trait. And we know that with Mendelian genetics, there are different variations of a gene, and we call those alleles. So here's our purple flowers. We got a white flower and a purple flower. We got a gene for flower color, and the two alleles would be purple or white. So that's Mendelian genetics. One gene, two alleles. One is dominant, purple. One is recessive, white. You inherit an allele from each parent, and that these alleles sort independently during gamete formation. Yes, there's a fourth one, but I'm not going to go into it because this actually doesn't work most of the time. We'll just stick with these. Okay, wait, did I mention something about Thomas Hunt Morgan being, I think, the father of modern genetics? He even said it took a bit of hammering to get out Mendelian thinking out of his students that one gene, two alleles, dominant, recessive, they're all on different chromosomes, inherited independently of each other. Yeah, that inherited independently of each other, no. He studied fruit flies at the turn of the century. And believe it or not, Thomas Hunt Morgan wasn't even that big on Darwin's theory of evolution by natural selection in the 1890s. He, of course, changed his mind. But these guys really expanded our knowledge of heredity in the early 1900s, including his students. And they all went on to go earn Nobel Prizes. They were amazing. And he studied fruit flies. And some of the biggest discoveries came when they discovered the white fly mutation. And these guys, they discovered that the chromosomes are the basis of inheritance, that your genes are on a chromosome. They discovered that mutations are the source of variation. That's amazing, right? It's mutations. That's your source of variation. They discovered that. They also discovered that there's sex-linked genes. So like color blindness in us, that's a sex-linked gene. Males get it because it's on the X chromosome. Yeah, so if you got one copy, you're going to express it. And they also mapped genes. See, genes have a specific place on a chromosome. It's called a loci. They mapped it. Created the first gene maps. It's amazing. And even to this day, we still use fruit flies as a model organism to study genetics and development too. Yay for common ancestry, right? I know it. It's hard to believe we have an ancestor with those guys, but we do. Might have to go back 600 million years, but hey, these things have been conserved for a very long time. Okay, so that's our heredity. Now, you know, an individual cannot evolve, but a population can. So these guys, Sewell Wright, Ronald Fisher, J.B.S. Haldane, they came up with the field of population genetics. And this focused on genetic variation within populations. And of course, that genetic variation is based on the different alleles in that population and how common those alleles are or how rare they are. And this, established that populations are the smallest unit of evolution, because we define microevolution, as you'll find in the next chapter. Evolution is a change in allele frequency from one generation to the next, right? But that happens in a population. So your genetics are changing around based on these alleles and how common or rare they become. If one allele is becoming more and more common, you're getting evolution, right? Or it can become less common. That would also be evolution as well. So, these guys also showed that you could have continuous traits. This is Fisher. So think about like a continuous trait like skin color. You can go from pasty white to very dark. Something like skin color, which is a continuous trait, is caused by many genes roughly following Mendelian inheritance. And of course, this shows the natural selection can alter these gene frequencies. They can change these allele frequencies in a population. So you could imagine if you take somebody that's, dark skin and put them up north where dark skin is actually bad because of vitamin D production, then the population over time might evolve to be lighter skin. Vice versa. Take people from the north where they have lighter skin because they get less sunlight, they have more vitamin D production. Put them on the equator where there's 12 hours of sunlight all day long. Over time, the population will evolve to be darker and darker and darker. So we're changing the allele frequencies to produce more and more melanin. That would be evolution. And of course, here's Ronald Fisher. Some really interesting thing about him. Might be one of the most important statisticians ever. So he developed the ANOVA, maximum likelihood, and F distributions. I mean, the guy was brilliant. We're not going to get into some of his odd cultural thinking, though. And of course, I love JBS Haldane, and I love this image of him, from before he lost all of his hair and went bald, because it happens to some of us. JBS Haldane is famous for getting asked, what he might be able to infer about the creator based on his knowledge of biology. He turned to this cleric and he said, God has an inordinate fondness for beetles. And that's, of course, referring to the diversity of beetles in this world. Beetles are one order of insects, and there are more species of beetles than there are vertebrates in the world. Fish, mammals, birds. Yeah, it's pretty crazy. Now, here's Ernst Mayr. Ernst Mayr only died like 16 years ago, so he was still around when I was in graduate school. And he was one of the first people to define species, an evolutionary definition of a species. And you're familiar with it. It's a biological species concept that basically a species is a group of individuals that can reproduce with each other. And I mean, the entire Endangered Species Act is based on the biological species concept. This is something I will talk about in later videos. He also came up with allopatric speciation. That's where you take a population, you separate them physically so you can't get gene flow. You can't have them interacting with each other, and they would over time diverge. So Ernst Mayr was a giant in biology. And then, scientists sometimes need ways to popularize their findings. And of course, Julian Huxley. Wait, haven't I mentioned Huxley? Huxley, does that name sound familiar? Well, it should. Thomas Huxley, who defended Darwin's theory of evolution, was his granddad. And Aldous Huxley, his brother, wrote A Brave New World. And it was Julian Huxley that termed the coin modern synthesis. And of course, Dobzhansky really put all of this together. He argued about mutations being the source of variability on a chromosome. And he used all the math of Fisher and Sewell and Wright and put it all together to combine it into one big theory. And he also at the time came up with other ways that species can evolve, or populations can evolve, through something called genetic drift, which is not natural selection. So he was another giant. And the point here is that the modern synthesis really shows that it took the work of a lot of people over decades to come up with this holistic view of evolution by combining Darwin's theory of evolution by natural selection with heredity and population level thinking. It didn't occur all at once. It took decades, and it took the works of all of these people to do that. So when we talk about evolution, when we define evolution today, it comes from the modern synthesis. And it's basically a change in allele frequencies in a population from one generation to the next. So what we're talking about here is, if we say a species changes over time, well, what's changing? The genetics, specifically the alleles, these variations of the genes. And we say that natural selection is causing these changes to occur. You can imagine if a disease hit somebody with A blood. Well, then in the population, people with A blood might become more rare. And then O and B would become more common. That would be an example of a disease causing evolution in that population. And of course, it happens over time. So there you go. And we use Hardy-Weinberg equilibrium to test for evolution at specific loci, or for the entire population as well. All right. So this is fantastic, right? By the 1940s, we had solidified evolution and genetics and heredity as our mechanisms of how species change over time. And of course, we came up with other mechanisms that would cause species to change other than natural selection. And we also explain how we have the persistence of genetic variation as well, because mutations are continuous. They occur every generation. So our current research, we didn't just stop at the modern synthesis. I mean, wow, that was 80 years ago. And so now we're starting to understand the effects of climate change on species. We're reconstructing the Earth's past to understand how species changed over time. And then there's other things that we've discovered. Reconciling horizontal gene transfer, endosymbiosis, epigenetics. I mean, lateral gene transfer. I mean, bacteria are exchanging genes all the time. And bacteria are not the only ones that are influenced by lateral gene transfer, also known as horizontal gene transfer. See, vertical gene transfer is from parent to offspring. Horizontal is you pick it up from the environment. And apparently about 5 to 8% of your genome is of viral and bacterial and other organisms' origin. It didn't all come from your parents. Well, I mean, in the history of our species, we've accumulated these additional genes that have actually influenced us. So for example, like the evolution of live birth in mammals, I love this example of synectin, which is a protein that allows the placenta to embed in the uterus, came from a virus. So one of the most mammalian traits, which is live birth with the placenta, may have been influenced by a viral infection that occurred 200 million years ago. Crazy. And then, of course, endosymbiosis. This is clearly not Darwinian evolution because it occurred when two organisms merged, two prokaryotes merged, and that, of course, triggered the evolution of eukaryotic cells. And I've got lots of videos on that. And then, of course, this whole idea of epigenetics. Lamarck was not as wrong as we made him out to be. Now, of course, epigenetics does not a change in the sequence of your genes, but it is a change in how your genes are expressed, which can be passed from parent to offspring. It happens all the time inside of our cells as part of our cell differentiation. But hey, if you're passing it to your offspring, if you're changing how your genes are being expressed, that's going to affect how your offspring interacts with its environment and it's going to affect its survival. Yeah. So what do you do here, right? I mean, how do we reconcile this? So these are where evolution is going now. We're moving beyond the modern synthesis and we're trying to reconcile this idea of lateral gene transfer, epigenetic inheritance, along with endosymbiosis into an even more complete idea of evolution, because at the time in 1940s, we knew of none of this stuff. Okay, well, I hope you enjoyed this modern synthesis. It's pretty amazing. And until next time, and my next video will be on the evidence of natural selection. We got a lot. All right, it's Dr. Kennedy on Tom Kennedy Science.